Hello, everyone. My name is Daria. I'm an account manager at Novogene Europe, and I will be the host of this webinar. I'm delighted to see so many of you were able to join us today. And today, my colleague Ruyan will give a presentation titled Maximizing Genomic Insights, an in-depth look at long-read sequencing. But before that, let me briefly introduce our company. Novogene was founded in 2011 and has been growing rapidly since then. We are continuously developing our portfolio of NGS and third-generation sequencing, and today we are present with services in various fields of science, from research, oncology, to food and water testing. Novogene is a global company with local contact persons and sequencing facilities in Europe, Asia, and the US. We have over 2,600 employees and more than 5,600 customers. Our multiple sequencing facilities equipped with Illumina, PacBio, and Oxford Nanopore technology gives us the largest sequencing capacity in the world. Our first European sequencing center is located in the UK, in the Cambridge Science Park. The state-of-the-art lab is equipped with Illumina technology and our patented intelligent sequencing platform called Falcon 2. The automated workflow of Falcon enables us to decrease turnaround time, minimize the factor of human error, thereby yielding consistently high-quality outcome. Thanks to our supercomputing capacity, we can process large amounts of data. As you might have noticed, we have recently launched another sequencing facility in Europe, specifically Germany. The German facility is gradually becoming more operational. Get in touch with your local contact person at Novogene to receive the latest updates relevant for your upcoming projects. We provide a full range of next-generation sequencing services: human, plant, animal DNA sequencing, various types of RNA sequencing, metagenomics services, and epigenomics services. In reality, our service offers extend beyond what is mentioned on this slide. We offer oncogene panels designed for solid tumors or hereditary cancers, and also accept pre-made libraries for sequencing. There are many reasons why you should choose Novogene as your sequencing partner. Providing consistently high-quality services is our priority. To achieve this, we perform multiple quality controls. There are sample quality control, library QC, and finally, data QC. We have a large capacity, which we are increasing all the time. Recent example of this is opening our German lab facility and also investing in the newest sequencing technology with improved throughput. Our patented Falcon 2 technology has not only a positive impact on quality and capacity, but also turnaround time, since Falcon can run 24/7. We offer customizable end-to-end solutions. This means we can process your project from extraction to analyze data, but you can also send us extracted DNA or RNA or pre-made libraries and choose to only receive raw data. Our product management team and laboratories are continuously optimizing protocols and services to ensure we have a sequencing solution that fits your needs. In that regard, we offer a range of platforms to suit your sequencing needs. Illumina for short-read sequencing, and PacBio and Oxford Nanopore technology for long-read sequencing. We are very proud to support researchers worldwide in their work, and today have a clear record in the scientific community. We have been cited in over 16,000 research papers with a total impact factor of more than 4,090. We are also driving the NGS industry forward with more than 60 NGS-related patents. Your contact persons are present in almost every European country. Get in touch with your local representative today to discuss NGS solutions for your projects. You can contact them directly or reaching out via our website. We also regularly host seminars in universities and various institutes across Europe. If you would like us to organize a seminar near to you, please do not hesitate to get in touch with us. I'd like to briefly introduce to you our talented European sales team. They are based in different regions across Europe and function as your first person of contact to Novogene. We speak your language. do not hesitate to contact us with any next generation sequencing related requests. Thank you for listening. That's all from my side for now. Ruyan, over to you.
Thank you, Daria. I hope my slide is presenting okay. Okay, great. And welcome everyone for today's webinar. The topic for today's webinar is maximizing genomic insight, an in-depth look at long-read sequencing. My name is Ruihan Bai, and I'm the product marketing manager for Novogene Europe. In today's webinar, we'll be covering the following content. First off, we'll be introducing the long-read sequencing platform. We'll be talking about how it works and some key parameters. We will also be introducing the application of long-read sequencing in different in genome, transcriptomic, and also metagenomic researches. In the last section, we will be sharing some tips and experience to carry out a successful long-read sequencing project. So let's start our webinar today with the evolution of sequencing tool. So in early 1970s, Frederick Sanger and his colleague developed Sanger sequencing. So it is a chain termination reaction to determine the sequences of DNA. Sanger sequencing became a golden standard, and it remained so for several decades. However, it still suffers from some limitation. Each time, the Sanger sequencing can only read around 500 to 1 kilobases, and it's relatively slow and also expensive. At around 2000, the next generation sequencing emerged. The platform including Illumina, Roche 454 pyrosequencing, and Torrent, etc. And the sequencing by synthesis technique has significantly increased the throughput of DNA sequencing and enable us to faster, cheaper, and providing a more detailed DNA sequencing. However, this second generation sequencing, it also have some limitation. For applications such as de novo assembly and some large structural variants, the application is limited. And then it is the topic for today's webinar, the third generation sequencing or the long-read sequencing. So the platform that features Oxford Nanopore and also PacBio. This new technology emerged at around 2010, and they are able to sequence native DNA in real time with single molecule resolution. So back when it was first invented, it has relatively lower accuracy, but this technology has also evolved itself, and at the moment, it's offering significantly improved accuracy. So you may ask, why should we choose long read? So if we imagine the question that we want to work out in biology as this jigsaw game, with smaller pieces, it's going to be very difficult. But if we are using larger pieces, we will be able to answer those questions not only easier, but also more reliable. So now we have a happy Dr. Gino. So without further ado, let's talk more about the platform for long-read sequencing. We'll be talking about how does it work and some key parameters. And we will start from PacBio. So smart sequencing, single molecule real-time smart sequencing from Pacific Bioscience, it is a novel technology that offers long-read and high accuracy sequencing. And you can see this is how the PacBio SQL series and also the review platform look like. The major advantage of PacBio sequencing is the superior accuracy. You can see that the read length is around 10 to 20 kb, but the accuracy is comparable with short read, if not better. And in this bottom panel, you can have an idea of how does the smart sequencing work. So this DNA polymerase is incorporating the NTP to this DNA template, which is the DNA that we want to sequence. The DNTP is linked with the fluorophore, And during the DNA synthesis, the fluorescence will be emitted and the fluorophore will be clipped off and leaving the DNA to be synthesized, being completely natural. The reaction and base callings are happening in this little nano well structure or ZMW, zero mode waveguide. So this is a special structure that enable real-time observation of single molecule and without the interference from background fluorescence. And so in this slide, we will see how DNA became sequences through this PacBio sequencer. So after DNA extraction, the high molecular weight DNA will undergo some mechanical DNA sharing. And then we will perform some DNA damage repair. This is important. Otherwise, DNA damage can lead to a prematurely, like leading to a polymerase early drop-off and affect the yield of sequencing output. And after that, there will be a SMRT adapter ligation. So this is to create a circular structure of the PacBio library. We'll also undergo a nucleus treatment to get rid of all the unwanted product. And after some cleanup or set selection, the library is ready for the PacBio sequencing. And we will talk more about this circular structure, circular library structure, because it enable circular consensus sequencing to generate hi-fi data. This is because the DNA polymerase will be able to read this circular structure over and over again and generating consensus data. Because the sequencing error is random, the consensus rate will cancel out the random error and giving data of very high quality. And in this panel on the right, you can see this is what happened inside the PacBio sequencer. So the PacBio flow cell, it is a chip with millions of ZMW. Typically, for a successful PacBio run, there are around 50 to 70% of ZMW that has one library and generating high quality data. And this picture is showing what is happening inside one of the ZMW among the million of them. So as we have introduced, the fluorescence will be used for base calling, and that gives the sequences of the DNA. And another aspect, because here we are talking about sequencing native DNA without any PCR amplification. Another aspect is the DNA modification. And PacBio is extracting the DNA modification through this nucleotide incorporation kinetics. To put it in a simple way, when the DNA is modified, when the DNA is, say, methylated, the incorporation of this nucleotide will be slower. And based on some machine learning and model training, the PacBio software will be able to extract the DNA modification out of this base kinetics. So now let's have a brief summary for PacBio. And here we are using PacBio Review platform, which is the latest, platform, and also it is the PacBio long-read sequencing with the highest throughput. So the smart cell is Review smart cell, which features 25 million ZMW, and the hi-fi yield per cell is around 90 gigabase. And here at Novogene, we are proud to share with you that the yield record is 135 gigabase hi-fi data per cell. So here, all the statistic is based on the current chemistry, but things may change soon due to the update from PacBio during the ASHG. In terms of sequencing runtime, the runtime is flexible, and you can run it up to 30 hours. In terms of run mode and base quality, PacBio Review uses CCS sequencing mode and generate hi-fi data. In terms of base quality, we can check this figure on the right. So you can see that the PacBio Review, the hi-fi data is generated based on this Q20 filter. Different projects may have a different distribution, but you can see that the majority of the data has at least Q30 accuracy. So that is actually a very good quality for sequencing rate. In terms of methylation, PacBio Review will be able to deliver 5-methylcytosine CpG site directly off the instrument. If you are interested in other type of methylation, such as non-CpG 5-MC, 4-MC, and 6-MA, you will also be able to extract those information based on the kinetics, as we have just introduced. Briefly, the advantage of PacBio sequencing, it is high accuracy, and is a very powerful tool for genome, epigenome, transcriptomic, and also metagenomic research. Now let's have a look at another long-read sequencing platform, Oxford Nanopore technology. So Oxford Nanopore technology, it is a revolutionary sequencing platform. It features a series of different sequencer from very portable devices that you can carry it with you everywhere to the industrial level platform. So the Nanopore, as the name suggests, it is a rate sensor chip with this Nanopore channel located in it. So this is a electric resistant membrane, and when forcing an electric field, the Nanopore will be able to record the disruption of the electric current bring by this nucleic acid strand passing through. So you can find that the Nanopore technology, it is independent of DNA polymerase, and therefore you will be able to not only sequence native DNA, but also you are possible to sequence native RNA, or maybe in the future you can even sequence protein with Nanopore technology. And because of this feature, Nanopore has some other very intriguing function, such as it is able to generate ultra-long DNA reads. And another interesting function is that you can employ Nanopore for adaptive sampling. So that means that you can achieve targeted sequencing, but without a prior enrichment for the targeted that you are interested in. And in this slide, we will use DNA, genomic DNA, as an example to illustrate how the Nanopore technology turns DNA into sequences. So we start from high molecular weight DNA, and it will also undergo some optional fragmentation or set selection, and there will be some damage repair and the adapter ligation. But difference here in this step is that the adapter contains a motor protein. We can see this figure or this little video on the right that the motor protein will dehelix this double-stranded DNA, and it will also work as a molecular brake. So it will control the speed of this DNA strand passing through the pore to achieve a better accuracy for the final base calling. So the disruption of electric current will be recorded, and this will be used for base calling. You will be able to extract the base sequences, and also the base modification out of this raw data readout. So in terms of base calling, you can either run it along the sequencing, or you can also elongate the time of base calling to achieve a higher accuracy. But of course, the higher the accuracy you require, you will need to put in more time and also more computational resources. Currently, Oxford Nanopore technology is supporting this Q20 plus chemistry. So it is the R10.4.1 flow cell coupled with this E8.2 motor protein. Collectively, you will be able to get a model raw rate accuracy at around Q20. Again, let's have a brief summary for Oxford Nanopore technology. We will again using Chromosome platform, which is the Oxford Nanopore sequencer with its highest throughput, and it is also the one that we feature in our Novogene lab. So it's supporting this promise on flow cell and supporting a Q20 plus chemistry. The theoretical maximum yield per cell, it is up to 290 gigabase. In terms of sequencing runtime, it is flexible, so you can monitor the sequencing all the time for the yield and also the core occupancy, and you can run it maximum around 72 hours. In terms of the base quality, with this Q20 plus chemistry, you'll be able to achieve a model raw rate accuracy of around Q20. For methylation, for DNA, you will be able to extract 5-MC and 5-HMC, and for RNA, you will be able to extract M6A under this DRACH context. So in summary, the Nanopore technology is having some interesting function, such as ultra-long capability. And this can be a very powerful tool when you want to, say, study the telomere to telomere assembly or T2T assembly, or you are trying to study a very big stretches of structural variants. Another interesting function is to sequence native DNA and native RNA sequencing. So this is a very powerful function when you are trying to study RNA methylation. So in summary, both PacBio and Nanopore platform share some similarity, but both of them have its unique feature and advantage. And here at Novogene, we are a certified sequencing provider for both platforms, and we proudly features 2 PacBio Review sequencer in our Germany lab to support your research endeavor. So after a brief introduction of the long-read platform, now let's have a look at just how powerful the long-read can be for different applications. We'll start from genomic research. So let's start with human genome. Over the past decade, we have learned a lot about our own genetic code. However, say clinical diagnostic, the short-read whole genome sequencing can only explain for a small proportion of them. While with long-read, based on its enhanced variant detection, this has been improved. In one of the study, you can see that nearly 70% of the structural variant, it might have been lost by the previous research that solely using short-read data. The advantage of long-read, including its ability to sequence native DNA and also perform phasing the gene. So that can be a very handy tool when you want to have a comprehensive analysis of the complicated genomic regions, such as pathogenic repeat or the homozygous sample. And here you can find an example of the Fragile X disease. And this figure is showing the comparison with WGBS, whole genome bisulfite sequencing based on short read, and also this haplotype resolved HiFi genome. So it's around showing the different performance at the FMR1 region, the pathogenic repeat region. Also, in a recently a benchmarking platinum pedigree, which is built by Illumina, PacBio HiFi, and Nanopore data, researchers has added approximately 200 megabase of high confidence region, and is now including some like repeat and segmental duplication into this benchmarking results. So long-read data can help us with a comprehensive variant detection. And here list some of the common types of genome variant, such as SMRT, indel, copy number variation, structural variant, which is it's gaining more and more attention because it's altering the most, in terms of the number of nucleotides, it is altering a much larger proportion of the genome, and also short tandem repeat, where a lot of research is showing that an expression of this short tandem repeat can be pathogenic. In the figure on the right, you can see that the distribution, like the scale of different variant. So you can see that they span from one single nucleotide to a stretch of 100 megabase. However, long read can span many of them easily. So unlike human genome, where a map of the genetic landscape is already available, this is still quite difficult for many plant and animal without a reference genome with good quality. So the challenges here include, for example, the genome size, the heterozygosity, and also the portion of repetitive sequences. And another aspect is that to provide meaningful insight, the reference genome should be of high quality. And the parameters can be classified as contiguity, which means that you want longer sequence, but also fewer of them in terms of number. And you also want to make sure the assembly is complete. And you can use some analysis such as boost code using the core gene or using some KEMA content analysis to check this parameter. And another very important parameter is the correctness. So you want to make sure the genome assembly you obtain is correct. And to check that, you can sequence the same sample with maybe short read data and check the SNP and also check the quality value directly. So how do a collection of DNA reads become genome? So after acquiring the scattered genomic DNA sequences, just like the jigsaw game we were talking about, we will use overlap to assemble them into context. So this is with long read, people are mainly using the overlap layout consensus algorithm, which is outperform the debut gene the algorithm, which is mainly used in short read. After obtaining context, by joining the oriented context, we are obtaining the scaffold. With additional sequencing inputs such as Hi-C, which is chromosome conformation capture sequencing, we will be able to bind the different scaffold into chromosome based on the strength of the interaction. And at this step, we are having a reference genome with a decent quality. But if we look closer, there could still be some gaps in the chromosome. As we have mentioned about the Oxford Nanopore's ultra-long capability, with Oxford Nanopore ultra-long data, by ultra-long we mean raw rate N50 at around 50 kilobases to even 100 kilobases sometimes, you will be able to close the gap and achieving a gapless chromosome from connecting from one telomere to the other telomere and resulting in a fully phased genome. So this is called T2T assembly, and this T2T assembly is setting a new standard for a complete genome assembly. So now with the reference genome in hand, we can explore about the unknown species or deeper our understanding for the existing ones. For example, you can perform whole genome sequencing. You can use it to perform whole genome sequencing to study the variant, or map it to your transcriptomic data to study about the gene expression pattern. Or when you have a collection of the reference genome of the same species, but exhibiting different traits, you can start to building a pangenome out of it, and also the phylogenetic genetic tree. It will be helpful to provide some diversity insight or study the evolutionary pattern. So this is done by this core and pan gene. The concept of core gene is that the genes are shared by all of the organism in this pangenome. So they are believed to be evolutionary important. And for the unique genes, for the pan gene, they only appear in some of the accessions. and therefore they are believed to play some role in adaptation to different environment. And let's delve deeper into this topic with a case study from Nature Genetics on this pearl millet pangenome. So pearl millet, it is a cereal crop with with a very good heat tolerance. So as our planet is getting warmer and warmer, the global warming is actually decreasing the yield of a lot of crop. And therefore, in this study, the researcher is investigating pearl millet and its heat tolerance with this graph-based pangenome method. In this study, they identified around 400K structural variants, and they showed that the expression of this transcription factor family and also the involvement of endoplasmic reticulum gene in the heat tolerance. So when overexpressing this RWPRK gene, it improves the heat tolerance, and it also transactivates the ER gene. The structural variant is affecting gene expression related to the heat tolerance, and also those near ER gene has shaped the crop adaptation. Therefore, this paper is offering some insight for creating a more climate-resistant crop. So this is actually a very interesting paper. In this paper, the researchers used de novo genome assembly to build the reference genome of 10 pearl millet sections from across the world based on the phylogenetic tree. And then they based a graph-based pangenome and identified the structural variant from this pangenome. And they are also using a multi-omics approaches, such as using RNA-seq and the enrichment analysis to identify the molecular mechanism behind the heat tolerance for this pearl millet. And we are proud to share with you that Novogene collaborate with the researchers in this paper, and we offer the library preparation, sequencing, and also data analysis for this work. And this is if you search pangenome in PubMed, and you can see that it's getting more and more popular. And in this slide, we will be showing some of the interesting findings from this paper. So first, this is the building of the graph-based pangenome. Here it is the Sydney plot. And this panel is showing the core and pan gene for the different accession. The core gene is shared, appear in all of this accession, and the private gene is only show up in a certain one of them. And the dispensable gene is shared by some of the accession, but not all of them. And based on this pangenome, the researchers is identifying structural variant, and they are doing some statistic and annotation for the structural variant. So they examine the types of the structural variant. So this PAV means present and absent variation. So it can be very useful when we are trying to study the adaptation to different environments. Another interesting figure is showing here. So the researcher found that a lot of structural variants, actually, they appear in the repeat region. So we can understand that with short read, it will be very difficult to achieve a high resolution when we want to study a certain the pearl millet genome. And they also annotate the structural variants based on where does they appear. So only a small proportion of them is showing in the gene. But that also makes sense because gene only account for a very small proportion in the whole genome. In this research, they also use multi-omic approaches to study the molecular mechanism behind the heat tolerance. For example, they check the differential gene expression, expressed gene, and structural variation to check the overlap. And they also check the expression, the influence of structural variation leading to the gene expression. So the next application we will be talking about is transcriptomics research. So long-read data will help us to capture full-length transcript easily. One of the difference between genomic research and transcriptomic research is the variability in transcriptomic. So this variability can arise from the abundance of gene expression and the complicated alternative splicing resulting in this complicated isoforms. So both PacBio and Nanopore, Oxford Nanopore technology can use poly-A enrichment to capture the full-length transcript. And so with full-length transcript, you will not need, you don't need to assemble the transcript anymore. And this is giving you a complete view of isoform. The long-read transcriptomic will help you to reduce the transcript fragmentation and improve the isoform detection. Also, it will help to increase the accuracy of detecting alternative splicing event. It can also characterize better novel and fusion transcript, and you can even study the abundance and quantification for transcript. We will talk about that in later. And now let's see a case study. So in this paper, the researchers is developing a bioinformatic tool based on Illumina short read and also PacBio isoseq to be more cost-effective, to provide a cost-effective transcript profiling tool. To study transcription profiling with short read, there are a lot of available tools, but each of them have some of the limitation. For example, the software like Histad2, it depends on a high-quality reference genome. And for the de novo approaches, they will use assembly, but sometimes they might result in a chimeric or wrong assembly. And therefore, in this paper, the researchers is developing a novel workflow. They first assemble the short read and perform a deduplication, and then they integrate with PacBio isoseq data. And in the end, they reassemble the remaining fragmented reads to create a final transcript. And the product will be able to use in annotation as a downstream analysis. And in this figure on the right, you can see that this workflow is actually outperform many of the existing tools. So it's a very interesting paper, but it's offering a very cost-effective tool, but it still have some of the limitation. The researchers is using this hybrid approach because PacBio isoseq used to be quite expensive. However, things has also changed nowadays with this PacBio Canex kit. So what is Canex? The PacBio Canex kit, it is based on this MASIC method, which is multiplexed array sequencing. And it works by concatenating the smaller amplicon into a larger one to increase the throughput. Here's a schematic diagram, and you can see that normally this is the scale of insert size for PacBio sequencing. And when you are trying to sequence such as transcript or 16S or RNA, it will lead to a wasted capacity. And Canex kit then concatenated this fragment to increase the capacity, to increase the throughput, and it will be very helpful for characterizing full-length RNA, single-cell RNA, and also 16S rRNA to enable a large-scale studies at a resolution difficult to attain with short-read sequencing. And in this figure on the right, you can see that when couple the Canex kit with Revio platform, we will be able to get around 50 million hi-fi transcript out of one sequencing run. So that's how you can also use long read to quantify your transcript. And the next topic we will be talking about is the metagenomic. Metagenomic is a cutting-edge technique that bypassing the need of culturing every individual organism. So a lot of the research is using the short-read data. However, in one of the research led by George Winstock, the leader of human microbiome project, is showing that the different amplicon may have bias in the taxa that they can identify. In this figure on the right, it's showing in a more straightforward way that the color of each branch reflects the proportion of sequence within this clade that could not be identified into the species level. So you can see that the 16S full-length RNA sequencing is providing a real and significant advantage. So you can obtain more species level annotation and sometimes even reaching a subspecies level with this full-length 16S RNA sequencing. And so in addition to the approach of using a marker gene such as 16S rRNA, another approach to study metagenomic is to use shotgun library preparation. With short-read, the workflow is like assemble into contig and bin them and getting a draft quality MAG. MAG is metagenome assembled genomes. And using this draft quality MAGs for the downstream analysis, such as taxonomy annotation, gene annotation, and also functional annotation. However, with long read, the assembly will be much more easier and the quality of MAG you obtain will also be greatly improved. You will even be able to obtain circular MAG from long-read metagenomic sequencing. And you will also carry out the downstream analysis with much higher resolution and accuracy. And now let's explore a case study. So in this paper, the researchers is using 16S rRNA gene sequencing to investigate the impact of the rapid urbanization. And in this paper, they choose a mega city in China, Shenzhen, and study the river and the coastal ecosystem. And they found that they use 16S rRNA sequencing and found that the pollution has disrupted the bacteria community in this ecosystem, and they also highlight the presence of some pathogens. So these findings are very important for managing bacterial dynamic and also for migrating the pathogenic risk in an urban setting. So the approaches in this study is the 16S rRNA sequencing by PacBio coupled with its CHAM2 ASV amplicon sequencing variant analysis with the CCS data. So there is some subtle difference between CCS data and HiFi data, but still both of them are having a very high quality, very high accuracy to give a reliable taxonomy annotation. And some of the results from this paper is shown on the right. So this is a taxonomy annotation you can obtain from this the 16S rRNA sequencing, full-length RNA sequencing, and also the researchers extract the microbial interaction out of the result. So having introducing the sequencing platform and some of the applications and the advantage, now let's share some tips and experience on carrying out a successful long-read sequencing project. So we will start from the DNA input. As we have introduced, long-read sequencing is a very powerful tool, but also it has much higher requirement for the input material. One of the reasons is because they sequence native DNA without any PCR amplification. And the other reason is that it is a very sophisticated technology and it's more prone to the interference from purity and also the integrity. So here we list some of the QC parameters and the importance of it. In terms of concentration, you will ensure accurate quality control and also to ensure to support a smooth library preparation processes. For the total amount, like we have introduced, because there is no PCR amplification, and also during the library preparation, like we have introduced, there will commonly has a set selection step to produce optimal fragment profile for the optimal sequencing output. And therefore, the total amount is also much higher than short-read sequencing. And the purity is another very important parameters because they may interfere with the sequencing reaction and lead to a decreased output. For fragment size, as the name suggests, long-read sequencing is generating longer DNA, and it's very important to have a high enough size because you cannot sequence what you don't have. DNA integrity. It's also an important parameter. So we can recall the library preparation processes where we have damage repair. Because those DNA damage, it can prematurely halt the sequencing processes. But with that being said, the DNA quality should still, the extraction processes should still minimize any possibility for this DNA break and damage. In the figure on the right, there's an example showing good like DNA with good quality and DNA with some severe degradation. This is under the pulsed field gel electrophoresis, which is a very high resolution tool to inspect the DNA quality. So after this webinar, we will share the slide with everyone, and there should be a clickable link guide you to our sample requirement website. But if you have specific question regarding your own project, you are also welcome to reach out to your local account managers. And in terms of high molecular weight DNA QC, we use qubit for quantification because this is less affected by the presence of contaminant. But we will also use nanodrop to check the purity of the sample. And we will also compare the concentration obtained by both of them because it shouldn't be too different. For the size and the DNA damage, we'll be checked with the pulsed field gel electrophoresis. As we have mentioned, it has very high resolution. And in our lab, we ran it like 17 hours. So it's able to properly migrate all the fragments inside the sample. Alternatively, another fragment analyzer you can use for long read is femtopulse from Angelet. In our lab, we use it for library quality control, and this is an automated pulse field capillary electrophoresis system. And in this slide, there are some tips and experience for successful high molecular weight DNA extraction. Due to time limit, I will not read them one by one, but generally to be gentle for your sample and to avoid any harsh chemical or vortexing them too much. So for a DNA with good quality, here at Novogene, we are also happy to guarantee the sequencing output for human, plant, and animal whole genome sequencing carried out with our Review platform. So in this table, you can see some of the listed guarantee for human and some mammal and plant. If the species of interest is not listed in the table, you are also welcome to reach out to your local account manager for more detail. So PacBio Review platform, as we have introduced, it will give you not only the hi-fi sequences, but also the methylation in one set of data. And at the end of the webinar, we will briefly share some bioinformatic workflow. We will start from de novo gene assembly. So for de novo genome assembly, we recommend to start from PacBio hi-fi data with at least 15x depth per haplotype. The assembler normally it is hi-fi assam, and this will give us a quantic level assembly. And when adding the high-C data, the genome scaffolding will give us a chromosome level assembly. So if you have higher requirement for the quality of the reference genome, you can also add the Oxford Nanopore ultra long data, normally 30 to 100x, to close the gap and reach T2T level chromosome. So after obtaining this de novo assembly, we can further perform the genome annotation. So we will annotate some of the repeat, the tandem repeat or the interspace repeat, and obtain a masked genome to facilitate the downstream annotation. We will be using the homology-based approaches and also the de novo approach. We will integrate the result with this EMV software and correct them with PASA. And finally, we do a functional annotation with different database. We will also be annotate the non-coding RNA, microRNA, small nucleic RNA, rRNA, and tRNA. And when a reference genome is available, we can also carry out resequencing services. And here I'm also showing a brief workflow. So after the quality control for the raw data, we'll perform a mapping to the reference genome based on minimap normally. And we can perform variant calling such as SV, SMP, indel, CNV, and short tandem repeat. And here I'm also listing some of the commonly used software. So due to time limit, I will not cover all of the services, but I list the services option here at Novogene for human, plant, and animal genome sequencing, microbial sequencing, and also transcriptomic sequencing. We will also be able to provide extraction services upon evaluation, and feel free to reach out to your local account managers. Finally, it is a summary for today's webinar. The take-home messages include long-read sequencing is addressing some of the limitation from short-read sequencing platform, and the major long-read platform include PacBio and Nanopore. So the advantage of long-read data in different applications, including enhanced variant calling, enhanced epigenetic detection, de novo assembly, capture full-length transcript, and also a comprehensive microbial diversity research. Last but not least, to carry out a successful long-read sequencing project, it is important to obtain the starting material with high quality. And that's it. And I'm happy to answer any questions.
Well, thank you, Ruyan, for this complete and comprehensive presentation. Well, we have received many questions actually, and because of the time limitation, I will go through a few of them. And the first question is, why are adapter ligation performed?
You mean the library? I think this question is about the library preparation for PacBio and Nanopore technology. So like we have mentioned, one of the advantage of this long-read platform is to sequence native DNA. And with this adapter ligation, we will bypass the PCR amplification and therefore enable native DNA or like native DNA sequencing.
Okay, the second one is, do you only offer the PacBio review sequencing for a whole run or flow cell, or do you also offer this service for lower levels of needed output, for example, 2 gigabases of output?
Oh, yeah, this is a very good question. At the moment for genomic sequencing, we are only offering the services in a full flow cell pattern. This is because the technology is sophisticated, and the sample within the same cell, they might interfere with each other. For example, the fragment distribution, and also the impurity, the carryover impurity inside the sample. But for RNA, for transcriptomic applications with PacBio, we are offering the package. So because there are some the PCR like amplicon cDNA generation, we are possible to use that on a per sample basis, and also full length amplicon. I hope I answered the question.
Thank you. Although you already mentioned during your presentation, I would like to still pass that question. Do all PacBio equipments provide hi-fi data? And what's the difference between CCS and hi-fi?
Oh, okay. So in this figure, there's another PacBio platform, SQL series. So it is able to generate it has two mode. You will be generating subarrays, or you can also extract the hi-fi data from the subarrays. So the difference between hi-fi and CCS, how to explain? I think it's kind of also rely in the software of the PacBio software update. For CCS, it is based on the passes. It is based on the passes of the rate, but hi-fi just filter. But hi-fi is a concept that's solely based on the accuracy score. So, but they are there's some subtle difference, but it's like how you define it. I hope I answered the question.
Thank you. And the last question would be, what is the limitation or weakness of PacBio and Nanopore? What's the overall error rate of sequencing?
Okay, maybe I will first talk about the error rate. So in this figure, it's reflecting the error rate for PacBio hi-fi sequencing. So actually, it's a very good profile. So all the data will have at least Q20 accuracy, and the majority of them is over Q30. And for Nanopore, you will be able to reach a model raw rate accuracy at around Q20. So for the limitation of long read sequencing, I would say accuracy is not much of a limitation for them at the moment. I would say cost is one of the limitation, and another thing is it's still kind of a complicated technology, and for example, during this sample requirement part, it has much higher requirement for the starting material because you will need a lot of DNA and it should not be contaminated, and you should also be very, very integrate. So that is some difficulty, I would say, for the long read platform. But yeah, but when I think PacBio is releasing their new chemistry and they mentioning the PR that they are reducing the starting material. So I think the technology is also getting better and better, and I'm very confident for that.
Great news. Thank you, Royan, for all the answers. Yes, thank you all for your time today. We hope you enjoyed the webinar and found it useful. Again, thank you for all your questions. As mentioned, if you haven't managed to answer your question live, we will get back to you via email next week with the answer. We will email you a link to the webinar recording and the slide deck. You can expect to receive it the week after. When the webinar finishes, a survey will pop up. We would appreciate you taking the time to complete this, as it helps ensure we make future webinars even more relevant to you and your research goals. As a token of our appreciation for your attendance at the webinar, Novogen is pleased to offer you a 15% discount on your upcoming project. An email will be sent to you within the next few days with a code that you can use to apply the discount. There will be a link in the follow-up email for you to view a certificate of attendance if you need one. This can be printed from your browser window. In the meantime, you can follow us on Twitter and LinkedIn to keep up to date with future webinars, offers, and news. And we look forward to seeing you all on the next webinar. I wish you all a great day. Thank you.
Thank you.